We found a new particle, the vault, and our first paper describing the particle was published in the Journal of Cell Biology. It was time to write a grant to allow us to investigate the significance of our findings. Would the grant reviewers think the vaults were worthy of funding? Hi, I'm Lenny Rome, the Vault Guy. I'm a cell biologist and a nanotechnologist. My passion is a common particle of the human cell called a vault. My goal is to explain what vaults are to non-scientists. In my previous 4 videos, I told you about the discovery of vaults, how they were named, and our efforts to publish the first scientific description of the new particle. If you haven't seen them, I'll put links below. Today, I want to cover 3 things. First, I want to tell you why grant writing is such an important skill for scientists. Second, I'll describe the process of applying for a basic biomedical research grant. And third, I want to return to the vault story and tell you where vaults are found in nature. Stick around. I think you may be surprised by how far back in evolution vaults show up. Our first paper about vaults described this new particle and suggested that vaults might be commonly found in many different types of cells. Here's an electron micrograph to remind you of what they look like. We were ready to submit a research grant focused on furthering our understanding of this new particle. Ultimately, it was aimed at figuring out what these strange particles are doing in the cell. My postdoc Nancy and I worked hard on writing a grant, and we sent it in and we kept our fingers crossed. For those of you thinking about an academic career doing research and teaching, you may have heard the expression, publish or perish. This might sound harsh, but in the field I chose, the biomedical sciences, publish or perish is absolutely true. Once you are fortunate enough to get an academic appointment, you need to raise research funds to support your lab, or you can't do experiments and you won't get data and you won't be able to publish. And soon you'll be looking for a new job. You know, a common misconception I hear from folks who ask me about my career as a professor at UCLA is when they say, "Isn't it great that UCLA pays you to do research?" Let me tell you, I only wish that was the case. Not only do I need to raise the funds to support my research and pay my staff, I also need to pay my postdocs and students who can't get their own fellowships. I also need to pay overhead to the university. Overhead is a lot like rent, and at UCLA, this charge is over 1/3 of every dollar that comes into the university from a research grant that I receive. In addition, except for the superstars, most professors at major research universities need to pay some or even all of their own salaries as well. Believe me, I'm no superstar. So just how does one go about getting a research grant? The National Institutes of Health, NIH, is the government agency that is the largest funder of biomedical research in the world. I introduced the NIH in episode 2 when I mentioned that I carried out my postdoctoral research there in Bethesda, Maryland. The NIH also runs a program that funds research outside the NIH campus. The total NIH budget this year is $45 billion, with over $25 billion going to research grants. This might sound like a lot of money, but there are way more scientists who want to get grants than there is money available to fund them. NIH reviews a variety of research grant types, including an investigator-initiated research grant called an R01. R01s are the most highly treasured grants for academic researchers to secure. Funding for this grant category is extremely competitive. Heck, there were nearly 8,000 R01 proposals submitted last year. The process of getting an NIH grant funded is complicated, however, it's recognized as fair. Allow me to explain how the process works. Grants submitted to the NIH are assigned to scientists who serve as grant proposal reviewers. This is a process called peer review. Peer review is also the process that operates when you submit a scientific manuscript for publication. I told you about this in episode 3. The grant reviewers are organized into groups called study sections. These are generally made up of around 25 to 30 scientists, organized around areas where their particular expertise is needed. For example, since there are always a number of grant proposals aimed at understanding cell structure and function, there is a standing NIH study section called Cell Structure and Function. If there are a number of proposals submitted to the NIH on a topic that isn't covered by an existing study section, then the NIH may form a new group. Each study section, which meets 3 times a year, may get about 100 grants in each session. Prior to the meeting, each research grant is assigned to 3 primary reviewers who read and write brief evaluations, called critiques. The remaining study section members get all the grants and they're asked to read them, but they are only required to write critiques on the grants that they are assigned. When the study section meets, the 3 assigned reviewers describe the grant and their critiques to the study section, and the entire group scores your grant using a scoring range from 1 being the best score to 10 being the worst. Yes, you heard correctly. As many as 30 scientists score your grant, but only 3 of them were assigned the grant to read and write a critique. After the study section meets, the scores of all the grants across all the study sections are normalized, and a percentile from 1 to 100 is generated that represents how your grant did among all grants during that particular review period. It can take 3 or 4 months after submission to receive your scores and your written reviews from the 3 scientists who are assigned as primary reviewers on your grant. In 1986, when our first Vault grant was submitted, an R01 grant would need to be scored in the top approximately 30% of all grants, the 30th percentile. This is to have any chance of getting funded. If you scored in the 20th percentile, you were in great shape. However, a score in the 35th percentile meant you were out of luck and sent back to the drawing board. Well, the process is even harder today, with less than 20% of grants being funded. Writing a grant is a lot of work that can take up a significant amount of time. Yet today, less than 1 in 5 grants submitted to the NIH gets funded. After waiting about 3 months, we received our scores and our individual critiques from the reviewers in the mail, which I passed around to the laboratory. Oh, I was delighted to see that the reviewers had really liked our grant and had given us an overall score in the 4th percentile. This was the best grant score I had ever received, and it remains to this day the best grant score I have ever received. Again, since this was before email had been invented, if you wanted to share documents with others in the group, I would need to make multiple photocopies. We did have copiers. To save paper and photocopying costs, I would attach a routing slip to documents that I wanted to share with members of my lab. Once reviewed, the recipient would check off their name and pass the document on to the next person on the list. When I circulated the glowingly positive reviews of our Vault grant to my group, one of the members of my lab scribbled a comment on the routing slip that stated, "Professor Rome's mom must have been reviewer number 2. The grant was for 3 years of funding, and we began by asking where vaults were found in nature, characterizing the vault components, and determining where vaults are found in the cell. These are the first things that any biochemist or cell biologist would want to know about a new cell particle. I only have time to tackle this first question in today's video. Recall in episode 2 that all living things can be classified into 2 broad categories, eukaryotic and prokaryotic. Prokaryotes, which include all bacteria, showed up on Earth first, and eukaryotes evolved from prokaryotes around 2.7 billion years ago. We looked for vaults in common bacteria, like E. coli, but we couldn't find them. Since we had found vaults in animals, which are higher eukaryotes, we decided to look for them first in different species of higher eukaryotes, and then in lower eukaryotes. Eukaryotes make up an extraordinary number of species and include all animals, plants, fungi, and many single-celled organisms like amoeba and yeast. A modern evolutionary tree of eukaryotes looks like this. The entire animal kingdom is just this one little branch. Let's look at a more familiar representation of an evolutionary tree. We were able to find vaults in mammals, amphibians, avians, and even in the lower eukaryotes, amoeba and trypanosomes, and in slime mold, a fascinating model organism. Finding vaults in lower eukaryotes was surprising and suggested that vaults might be ancient particles. Lo and behold, 23 years later, a research group at Massey University in New Zealand studied all the published vault sequences available, and they concluded that vaults were present in the last eukaryotic common ancestor, LECA. LECA is a hypothetical ancestral state referring to the first cell that gave rise to all eukaryotes on Earth. Here's a chart with the eukaryotic supergroups. They all have this theoretical common ancestor. Boy, this is very cool. The first eukaryotic cell that gave rise to all eukaryotes had vaults. It was also intriguing to us where vaults were not found. We could not find vaults in yeast, worms, most insects, and plants. This meant that during evolution, vaults were lost from these 4 eukaryotic organisms. You know, these organisms are some of the best biological systems for carrying out genetic studies. I used to tell my lab that nature had decided to make it really hard for us to figure out what vaults do for a living. The original antibody we had raised in a rabbit against rat vaults turned out to only bind to the major protein of the vault. As this antibody also reacted with similar sized proteins in all the different animal vaults we had characterized, we concluded that the vault particle must be highly conserved as well as broadly distributed throughout eukaryotic evolution. Such conservation and distribution suggested to us that the particle might carry out an essential function in eukaryotic cells. Little did I know, my favorite phrase, that elucidating vault function would become a lifelong pursuit. I'm Lenny Rome, the Vault Guy. Join me in the next video where I describe the individual components of the vault particle, which for such a big particle surprisingly only had 3 proteins. I'll show you our first model of the structure of the vault. Feel free to leave a comment below and subscribe if you want to see more of the vault story.